Hello students, this is Dr. Faizan Mirza. I'm discussing with you photosynthesis today. We need to understand what autotrophs are and what are heterotrophs. Autotrophs are organisms which can synthesize their own organic compounds, which means food, by using raw materials from the environment. For example, plants are producers of ecosystems in like all over the globe. What are heterotrophs? Heterotrophs cannot synthesize their own organic compounds, food, by using raw materials from the environment. Rather, they depend on prepared food, prepared organic food, and that's why they are consumers or decomposers. So we have to feed on some plant diet so that the nutrients that are stored in the plants become part of our cells, and we build our protoplasm from that. So we are heterotrophs. So autotrophs, which can synthesize organic compound from raw materials in the environment, they can be categorized as photoautotrophs or chemoautotrophs. Photoautotrophs are those which are using light energy to make their own raw materials, to make the raw materials turn into the compounds that they need, sugars and amino acids and proteins and lipids and whatever they want. And these include green plants and eukaryotes. Photosynthetic prokaryotes include cyanobacteria, it's a prokaryote. Prototists, which include the algae. These are eukaryotic unicellular organisms. For example, green algae, brown algae, red algae. All of these actually are photoautotrophs. What are chemoautotrophs? Chemoautotrophs are organisms which are microorganisms, and they have mesosomes. And mesosomes are enfoldings of the inner of the cell membrane, not the inner membrane, the cell membrane of the bacteria. And these mesosomes actually have the enzymes on which the catalytic action occurs, where they are able to catalyze a particular set of reactions, and from those reactions they gain enough energy to make their own nutrients. For example, nitrogen fixing bacteria, denitrifying bacteria, they all catalyze a specific reaction and they extract energy from it for synthesis of organic compounds using raw materials. This is how a chloroplast looks like, having double membranes, an outer membrane and inner membrane. There is an intermembrane space, but very thin. The fluid present inside the chloroplast is called as stroma. In stroma, you have lipid droplets, you have starch grains. The larger one is the starch grain. It would stain blue-black because of iodine solution. And these are just lipid droplets. So where the starch and lipid droplets came from, they are just how the sugars got converted into these compounds because of the chloroplast wanted to store it for a long time. These stacks, what are these stacks? These stacks are called as grana or granum. These stacks actually are made up of individual membrane enclosed compartments. Each part of this stack, it's like a stack of coins. So each part of this stack is a thylakoid. So there are various thylakoids, one on top of another. They together form one granum, and one granum is connected to the next granum through a lamellae. So there are several lamellae running all across the chloroplast, connecting these grana together. There's a low concentration of H plus ions in the stroma, and what's the significance of this low H plus ion concentration? We'll discuss this later. If you look at an electron micrograph, this is how the stack of thylakoid looks like. Each thylakoid is actually a membrane enclosed compartment, and they are all stacked on one top of the other. So this is granum. So if I ask you to label A and label B, B is the granum, or you can label this as thylakoid if it's singular, a single part of this stack. This is thylakoid, and if this is whole, this is granum. The fluid present outside the thylakoids, this must be stroma. So that's the stroma for you. When we're discussing structure and structure and function of chloroplast, as already covered, I'm not covering the details which I already have mentioned. The thylakoid are there. Thylakoid, what's the importance of thylakoid? Thylakoid are the site of light-independent reaction. Sorry, the light-dependent reaction. Sorry, not the light-independent reaction. This thylakoid is the region where the light-dependent reaction occurs. Light-dependent reaction means that this is exactly where the light energy will be harvested, and it will be turned into chemical potential energy, and this is where the food that will be started to be made, not the eventual product, but yes, it will start to be made here. On some reactions will occur here that are dependent on light energy. We'll discuss this in short time. So photosynthesis basically begins here on these thylakoids. What are the products of the light-dependent reaction of photosynthesis? We know that photosynthesis occurs in two stages. There's a light-dependent reaction and there's a light-independent reaction. So thylakoid is a region where the light-dependent reaction occurs. The light-dependent reaction, whenever it's occurring, there are three products of light-dependent reaction. In most cases, it's oxygen, there's ATP being produced, and there's reduced and ADP being produced. So there are three compounds which get produced in light-dependent reaction. Reduced NADP is similar to NAD. It's also a hydrogen carrier, as we have discussed in mitochondria, that there are hydrogen carriers. So NADP is also a hydrogen carrier. Now, there are ribosomes in chloroplast, and these ribosomes are 70th ribosomes, and these 70th ribosome can carry out translation for protein synthesis. The grana, the thylakoid, I just told you that this thylakoid is actually a membrane enclosed compartment. So this membrane enclosed compartment is having this membrane, this black line at the top. This is a phospholipid bilayer. This is a thylakoid membrane, and the thylakoid membrane, on the outer side of it, there's a stroma, and along the inside, there will be the thylakoid space present. So thylakoid space is this tiny gap between two black lines, which actually combinedly is a thylakoid. So this is the thylakoid space in the middle of the two black lines. So if we just try to visualize how that one black line or membrane looks like, so it's just a phospholipid bilayer. Just beneath it, you will have thylakoid space, and above the phospholipid bilayer, you will have stroma. This thylakoid membrane will have photosystems interspersed, and these are photosystems. This is not a protein, this is not a glycoprotein, it's a photosystem, a completely different kind of arrangement of photopigments, and this is exactly where the light-dependent reaction occurs. So if I draw this granum here, so this is having, for example, 3 thylakoids here. This black line, this black outline is the thylakoid membrane. This white region between two adjacent black lines is the thylakoid space. And these pink structures that I've drawn here are the photosystems which actually are present on the thylakoid membrane, and these are the region where the light-dependent reaction occurs. So, with that we just move on to looped DNA. There's a looped DNA as well, and that will have all the genes that the chloroplast will need. So we know that mitochondrial DNA is having every gene that it would need for protein synthesis. So it's having a complete genetic makeup. But chloroplasts, on the other hand, do not have a complete genetic makeup. They do have a looped DNA, which is having a lot of genes that chloroplasts will need for synthesis of various proteins. But yes, the chloroplast in turn will have to depend on the nucleus of the cell for extra genetic information, which this chloroplast will need to function. Then the stroma. What's the function of the stroma? Stroma is the site where the light-independent reaction occurs of photosynthesis. The light-independent reaction of photosynthesis is also called as Calvin cycle. In the Calvin cycle, sugars are produced, and reduced NADP is oxidized back to NADP, and ATP is hydrolyzed to ADP and PI. So you can figure it out from this that the products of the light-dependent reaction, which means ATP and reduced NADP, they are used up in the light-independent reaction, where the reduced NADP gets oxidized back to NADP, and ATP is broken down to ADP and PI, and in turn sugars are produced. So, for example, if we come to this diagram shows to you this is the chloroplast membrane, and these regions are labeled. If I ask you to label X, it would be the region which is the stroma. Y is your again thylakoid, and this entire structure is the granum. And W and Z obviously are the stromas as well. So if I just come to another diagram, this again similar to it, I think you can just label where these structures are present. So this question actually is not asking you about W and Z to like what W and Z are. This question is asking you where you need to label this letter W and a letter Y. Or where exactly is this happening? So, for example, W, use the letter W for the diagram wherever the light-independent reaction occurs. We know that light-independent reaction occurs in the stroma, so W must be labeled here. Then it's saying where chlorophyll is found. So we know that photosystems are present on the thylakoid membrane. So this is the region where the thylakoid membrane is present. To your point, the label should point precisely at the membrane here. Then this question here, label using a letter Z, the region where the reduced NADP is oxidized back to NADP. So this also occurs in stroma only because we know that this is where the Calvin cycle is occurring. Label using a letter X, the region where H+ ion is low in concentration. So we know that in stroma, H+ ion low in concentration. So that actually brings us to the idea that why I have used 4 letters here to just make sure that the question that are mentioned here are answered. These are just the adaptation of a structure of chloroplast according to their functional role. And that's it from my side. Thank you so much.